creating nanoscale force sensors using DNA origami, which can respond to the biological dynamics and forces between cells. Sounds cool, huh? So we thought. We want to create a nanomachine that enables us to measure things we have never been able to measure before. We were inspired by the idea that cancer cells have different characteristics than normal, healthy cells. These cells are unique in their unrestrained growth. Invasive cancers are known for their ability to grow and exert forces on other parts of the body, thus damaging organs far beyond the primary cancer site. The exertion of forces are familiar at macro scale. What we would like to develop is a tool that can measure forces at nanoscale, like those in the cytoskeleton of a dividing cell. This tool could be made using DNA origami, a technique that leverages the structural rather than the information storage qualities of the famously stable DNA double helix. By programming the sequence of DNA strands, we can precisely shape the exact tool for the job. Our tool measures force using a spring created from DNA strands. This spring will have a known spring constant derived from lab tests. The opposing ends of the spring can be bound to measuring sites in the cell. As these sites move and exert a force on the spring will stretch. The force required to achieve a certain spring deflection is correlated by Hooke's Faux law. When a specific force is reached, so is a specific amount of spring stretch, which in turn will release signaling molecules held within the tool structure. We can finely tune the tool to release this agent at specific forces by changing the required stretch. Logically, some ethical problems could arise. Injecting our DNA origami into the human body, which some people could see as a mini-robot, could lead to some resistance. Besides, patients could be imposed to unnecessary danger when our DNA origami would create a false response by reacting to normal healthy cells. However, we strongly believe that our DNA origami, when enough research would be conducted, could lead to great innovation in the medical world. DNA origami force sensors can be adapted to be used in a wide array of applications by customizing binding sites, critical release force, or signaling agents, promising to be an exciting opportunity doing fundamental research that leads to the creation of future therapies.
Opa!